Thank you very much, Thomas. I'm Stephen Smith. I was 35 years at Pioneer, working on intellectual property, genetic gain, and lots of genetic resource issues. I think it's important to remember that this year is the 50th anniversary of the National Academy Report on Genetic Vulnerability of Major Crop Plants. And if you don't know anything about that, you should look into it and get to understand why that report was necessary and what it had to say. So Peter Bretting is National Program Leader of Crop Production and Protection at USDA ARS. Peter, we're very happy you were able to join us. And I see behind you, you have the past and the future. So I'd like you now to give your presentation on about plant germplasm and data for breeding and research, and you're going to tell us about an app that that's now available to facilitate use of these genetic resources. So over to you, Peter. Thank you very much.
Thank you very much, Stephen. I presume that everyone can hear me. Can someone give me a thumbs up? Yeah, all right. Thanks very much. Good morning, and thanks to the National Association of Plant Breeding for inviting me to discuss the U.S. National Plant Germplasm System and its importance to plant breeding in the U.S. and globally. In addition to funding, staff, and the other items listed on this slide, access to genetic variation in germplasm is crucial to realize genetic gain through breeding. Most breeders, most of the time, depend on elite breeding stocks developed over decades through cycles of recombination, selection, and adaptation, but breeders of new crops, new market classes of crops, and those responding to new and unforeseen challenges might require genetic variation absent from their local gene pools. Fortunately, to use a common phrase now, there's an app for that in the form of the U.S. National Plant Germplasm System and its information management system, GRIN Global. Breeders expect that after placing an order through GRIN Global or communicating with an NPGS curator, healthy, clean, well-documented germplasm that meets their specific needs will be delivered promptly. You expect that curators will help identify the germplasm that's fit for purpose and which germplasm should be avoided. When authoritative information about the correct scientific names and potential uses of germplasm is needed, you expect GRIN taxonomy will provide that information. And to a large extent, those expectations are met. The NPGS distributes an average of more than 200,000 germplasm samples to fill an average of more than 10,000 orders a year. Over a recent 5-year period, that's 1.2 million samples distributed. 2/3 to 3/4 are to domestic US recipients, with the largest single category of recipients, the faculty and students at land-grant universities. And about 2/3 to 3/4 of the US requests that are filled are to the public sector, the remainder to the private sector. It's not a quota, it's just a long-term average. And plant breeders put that germplasm to good use. Here's just one example of germplasm's impact. A particular peanut accession was maintained in the National Plant Germplasm System for 25 years with little use before the discovery that it was highly resistant to a virulent virus. Breeders rapidly incorporated the gene into US peanut gene pools. And more than 20 years ago, the annual economic value of that trait was estimated at $250 million a year. Today, that value must be much higher. And that's just one trait from one accession of the now more than 600,000 accessions of more than 18,000 species conserved in the National Plant Germplasm System. So how can the NPGS app accomplish these achievements? Simply put, years of dedicated hard work. During this NAPB meeting, please seize the opportunity to tour one of the premier NPGS gene banks, the USDA, ARS, Iowa State University, North Central Regional Plant Induction Station. Since 1948, successive generations of this gene bank staff have conducted all the different operations listed here at the left, from acquiring new germplasm, maintaining thousands of accessions, increasing inventories via controlled pollination, managing millions of germplasm records, and of course, distributing samples. The intrinsic genetic variation is genetically characterized, and agronomically and horticulturally important traits are evaluated. The gene bank staff also works very closely with the genetic enhancement programs, particularly with the co-located germplasm enhancement of maize project, the GEM project, which you can also see during the tour. So I hope that you can become familiar with the NPGS gene bank while in Ames. But what about the NPGS's future? Keeping in mind the perils of forecasting, here are some thoughts about what is to come. It doesn't seem too hazardous that breeders and agriculture as a whole must adjust to accelerating rates of environmental and market changes, some of which could be revolutionary. For example, the massive volumes of information readily available, innovative supply chains, and other factors might increase the demands for more variety of foods and other agricultural products, thereby increasing the demands for cultivars with novel traits at germplasm. Therefore, we might expect that breeders and researchers will want more germplasm, more information and data about that germplasm, and more advice and recommendations from the curators. So how can the staff of the 20 National Plant Germplasm System gene bank locations, shown here on the USDA plant hardiness zone map, meet such demands? Well, first, the National Plant Germplasm System must deal with current challenges. In many cases, there are substantial mismatches between the responsibilities for adequately conserving and facilitating the use of germplasm and the knowledge and operational capacity to do so. As a result, sometimes substantial backlogs in germplasm management exist. Curators must balance many different competing priorities. Some of them can surely empathize with the scenario depicted on the right of this slide. Seemingly innumerable new biological, meteorological, and bureaucratic challenges must be faced, and plant breeders face many of these also. But if current and future innovations can improve gene bank operations, these challenges can not only be overcome, but substantial advances will emerge. One example is new technology from the Fort Collins Gene Bank to detect deterioration of seed quality, not via destructive germination tests, but rather via the rate of degradation of RNA in the seeds. The maize curators you'll meet in Ames have developed innovative, more efficient regeneration methods for teosinte, the maize crop wild relative that contains numerous potentially valuable traits for corn breeding. The Pulse curator and collaborators at the Pullman Gene Bank have developed a suite of new genomic tools that not only can contribute efficiency to pea germplasm management, but also accelerate breeding peas for new market classes in support of the expanding plant-based protein industry. The Pulse curator at Pullman, the hemp curator at the Geneva, New York Gene Bank, and their collaborators are adopting high throughput phenomic approaches to more rapidly, comprehensively, and accurately evaluate germplasm traits to generate valuable information for guiding plant breeding. During the Ames location tour, you'll see the close working relationship between the gene bank and the GEM. The gene bank provided the starting materials for this more than 25-year-old breeding project and curates and distributes the inbred lines that the GEM project develops. If additional support is available in the future, some gene bank staff may take the lead for conducting genetic enhancement projects with underserved crops. In fact, the amaranth and quinoa curator at the Ames Gene Bank is already addressing key traits, adapting unadapted germ populations, and broadening the genetic base for these crops, generating new breeding stocks for other plant breeders. In the future, the National Plant Germplasm System app will be expected to deliver a greater volume of more comprehensive genotypic characterization and phenotypic evaluation data. Through time, the quantity and quality of such data might become adequate for advanced search engines and artificial intelligence to enable automated replies to the many queries that gene banks receive and enable curators to gain valuable insights from latent patterns and connections in massive data sets. A crucial current and future goal for the NPGS is to capture the decades of experience accumulated by germplasm managers and curators and transmit it to the next generation. Following this presentation, my colleague Pat Byrne from Colorado State University will describe the training and educational project that the NPGS and collaborating universities have established precisely to attain this goal. So far, I've spoken primarily of what breeders and researchers expect and need from the NPGS. What does the NPGS need from you? Most important, it needs your continued support by communicating the importance of the NPGS to decision makers. Comprehensive documentation about the scientific and economic impact of NPGS germplasm and of the system is extremely valuable for this purpose. Strengthen partnerships through service on the 44 different NPGS crop germplasm committees, collaborations for germplasm evaluations and characterizations, and the research goals are all crucial now and for the future. After all, plant breeders and NPGS staff share the same long look, a focus on long-term goals and commitment to work now and over decades to attain those goals. We recognize that leading people, and especially fostering the development of early career colleagues, is one of the most important roles we can play to secure a better future. So in closing, thanks to the National Association of Plant Breeding for its continued leadership in raising public awareness of plant breeding, supporting early career plant breeders, and the NPGS, and for your attention. So with that, if there are time, I'll be happy to entertain questions.
Okay, thank you very much, Peter. We actually have over 10 minutes for questions. So let the people with the microphones know when you have a question, put your hand up. And if you get carried away before the microphone gets to you, you'll just have to repeat your question, okay, which is fine. I'm just going to alert you to the fact that Peter will need to listen to your question via a microphone. Okay, the floor is open.
Any question?
We have a question coming, Peter.
Okay.
You don't get to call a friend yet. Okay.
So Jamie Sherman from Montana State University. Curious, how does your funding work generally? So we would know better in how we could help support you in getting that funding. So who are the best people for us to talk to in support of you?
Well, thanks. Our support is through federal appropriations mainly, and that is determined by a combination of the administration's priorities and the power of the purse strings of the Congress. So at your universities, there are probably administrators who have as their job to communicate your university's priorities to decision makers, and that would be a good route for you to follow. Membership in organizations such as the National Association of Plant Breeders, American Seed Trade Association, the Tri-Societies, these are key organizations for communicating to decision makers your needs as plant breeders and the importance of the national plant germplasm system. So thank you for that question.
More questions? We have one here. Mario doesn't usually need a microphone, but even to get to Washington, he probably does.
Hi, Peter. Mario Carlone from Corteva. I was wondering, and maybe I don't completely understand the system, but evaluate any of the stuff that's in storage for new upcoming diseases at a, like, we'll just take tar spot as an example. It's relatively new and going into you know, a higher frequency, if you want to call it that. Does the organization try to identify some more recessions or accessions or stuff that might be of value fast in those cases so that people know where to go look?
Thanks, Mario. Crop germplasm committees that I mentioned during the talk are key to that. We learn about our curators, administrators, learn about these new challenges. Sometimes the first time we hear it is through a crop germplasm committee. That was the case with tar spot a few years ago. And once we learn of those challenges, if the resources, the support is available to plan or actually begin conducting evaluations of germplasm that might have a resistance or tolerance to such traits. During the tour, I think you'll find they try to keep very much connected with their crop communities to learn very quickly what some of these oncoming challenges might be.
Okay, more questions. Over there. You next.
Hello, Peter. It's Hannah Senior here. I wondered whether you could comment on the relative importance of communicating the importance of NPGS to politicians, political decision makers, versus others in the agri-food community, other corporate interests or other decision makers.
That's crucial, Hannah, for the question. Decision makers on the political scene know the importance of it, know that support is crucial, and it's a wise investment. It's well spent. It's, you know, some might say, oh, it's an insurance policy for the future. That's true, but it's insurance policy that's paying substantial dividends now, as the example that I cited for that one gene in one peanut accession and its value. And there are examples of that. What is very important and we find somewhat difficult to secure is information about the economic impact of the germplasm from the NPGS. Not surprisingly, corporate interests are quite reticent to divulge details of their breeding programs, the content of the materials that are in, or details of the content of the genetic variation that is used. But I'd like to think that there could be ways of communicating the importance, whether it's absolute or comparative importance of the germplasm to the economy US and global agriculture. But for that, one needs data, and sometimes those data are difficult to come by.
If I may just add to that, we heard from Andy Levine yesterday from American Sea Trade Association, and the ASTA with NAPB have been instrumental and are continue to be in taking a load of us up to the Hill and talking to Congress people and senators. And it's just totally astonishing, particularly in Congress, how little Congress people, even from understand about plant breeding and genetic resources. So every bit of education. Okay, over to you. Thank you.
Good morning, Diego Viteri from University of Puerto Rico. My question is, how effective are the new methodologies to evaluate the degradation in the seeds compared to traditional methods? For example, the viability of seeds or stuff like that. Thank you.
Thanks, Diego. These methods are still under development. They're very recently published studies I can provide you showing with soybeans, actually, showing the value of DNA, measuring DNA degradation for predicting future deterioration of seeds. You know, at the germination tests are roughly positive, negative, yes or no. It germinates or it doesn't. And they're not very good in many cases of predicting be near catastrophic losses of viability. In other words, you might have germ tests at 70 or 80% in year 1. In year 10, all of a sudden it's 20% or less. And that's the case for many species. What the researchers at Fort Collins have found that the relationship between loss of viability and deterioration of the seed and degradation of RNA appears to be almost linear. So one can, we hope with further refinement, be able to predict when will be absolutely key time points to regenerate those materials, rather than finding sort of ex post facto that, oops, we missed that since we test germination 10 years, we missed that at year 5, all of a sudden the viability is really decreasing.
Okay, thank you very much, Peter. We still have more questions, but we have no time. So I will ask anyone, all of you who have further questions, to contact Peter, and Peter is incredibly good at getting back to you on emails or via the telephone. So let us all give Peter a round of thanks for coming to us today. Thanks.